Okay, so switching now to the vaccines, we've been talking about vaccines so much more recently, and because of the pandemic and the urgency of the pandemic has led to this explosion in vaccination technology, in the sense that there are vaccines getting into large numbers of humans that have never been in this many people before. And so a lot of our vaccines, particularly a lot of new vaccines that have been made recently because of safety profiles and reactogenicity, were relatively simple vaccines that were based on recombinant proteins or some form of viral inactivation. So this is what the flu vaccine is normally every year. And you get the flu vaccine and most people don't have a lot of side effects. They don't even feel very bad. The two major vaccines that have been licensed for SARS-CoV-2 are the viral vector vaccines, which include the J&J and AstraZeneca vaccines, which use a defective adenovirus that's been swapped out with a piece of the spike protein. And then the RNA vaccines, which the Pfizer and Moderna vaccines, which are a lipid particle containing RNA for the spike protein. And so going back to our innate immune response, lipids, and particularly weirdly modified lipids, and RNA, and particularly weirdly modified RNA, are both great signals for pathogens, and your innate immune system responds to them. And this is in part why these vaccines have been so effective, and why those of you that have gotten them, sometimes you feel pretty bad, particularly after the second dose of these vaccines, which are really amplifying inflammatory responses. So one critical feature of thinking about vaccines and vaccine design, and potentially some of the differences that we're seeing in efficacy across the different vaccine platforms, is that even though all of the vaccines that you've seen come out in any country in any form have largely been spike-based vaccines, they're not all using the same spike. And so we talked earlier about how this is what spike looks like when it's normally on a virus surface, and when it binds to ACE2. So the RBD is up here. This is sort of the surface it's using to stick to the ACE2. And then once it's inside the cell, when it wants to fuse with that endosome and dump its contents out, it turns into this stabby structure that's going to poke a hole through the endosome and get out. And so lots of vaccines have decided that you don't want antibodies to this version of spike. You want antibodies to this version of spike. And there was a lot of work done on SARS-1 to figure out how to stabilize this protein in this form. And a couple of mutations were made, changing certain amino acids into prolines. And it stabilized this structure so that it wouldn't trigger into this structure. Because what happens for both HA and for spike is that what triggers this flip here is often a pH change. And so when you're prepping a vaccine or even just if you're talking about an RNA vaccine or a viral vector vaccine, and the vaccine's being made inside your cell, the antigen can undergo that pH change just under a number of different conditions. And so a lot of the protein that's exposed to your immune system looks like this. And so you'll make antibodies to something that your immune system will not ever really see. And so a lot of the most effective vaccines use what's called this 2P form of spike that's stabilized in this way. And specifically, Pfizer, Moderna, J&J, and Novavax all use the 2P form. AstraZeneca did not, and neither did any of the Sino vaccines. Okay. And this is in contrast to the way the annual flu shot works, as I mentioned, is we just take flu virus, we perform what's called a detergent split, where you add basically just a little bit of soap, you break apart the viral membrane, and then you reform sticky rosettes of the viral proteins and purify out complexes of the HA and NA with very little other components of the virus, and no adjuvant is added. And so this is probably in part why influenza vaccines are not particularly potent compared to what we're seeing for SARS-CoV-2. I've included some data in here that just for general interest on some of the early data from the phase 1/2 trials from each of these vaccines that's led to their eventual licensure. Again, most of them look very good, though they have different levels of efficacy. And really critically, it's important to look at the endpoints that were used to define that efficacy. No two vaccines have used the same endpoint to determine their efficacy. And so that's another difficulty, especially when you're thinking about modeling and comparing relative efficiencies. They're not actually using the same endpoint in terms of the symptoms that they're looking for, the way that they're screening, and the frequency of testing. And again, this is just more summary material for you for reference, just because I know this is topical for people. And I wanted to get to how you can start looking at some of these responses in terms of efficacy. So Pfizer was one of the first companies to report, and there's probably the most research on the Pfizer from an immunological perspective and an epidemiological perspective. And so the numbers really in the Pfizer trial and the phase 3 trial were that 43,000 individuals were in the study. They had 170 COVID-19 cases. 162 of those were in the placebo group, 9 of which were severe, and 8 were in the vaccine group, 1 of which was severe. And remember, at this point, they don't really know the timing of when protection is going to kick in. This is what the study was designed to look for. The Moderna data looked very, very similar, but this is what the incidence curves looked like for the two groups. And so you really saw early in the first period after the vaccination, this is after dose 1, the two curves are tracking each other, and then the vaccine curve here in blue just flattens out. And so this, has led a lot of people to say, really, by even day 7 or so after your first shot, you're probably protected from symptomatic or severe disease, because that was how Pfizer was catching these cases. Whereas the curve in the placebo group just kept going up, 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 up. And so, unfortunately, because of the speed with which we wanted to data, there's not a lot of good data yet on whether or not the protocol that we're using in terms of the 2 doses 3 weeks or 4 weeks apart is really ideal. There's some data now coming out of the UK where they actually force people to take the doses much farther apart to spare dosing, that might even suggest that dosing farther apart would be even better. As I mentioned, there were RNA vaccines previously, but none of them were in wide use in humans. So there were these experimental vaccines here, but you can see they were all still in phase 1/2 trials and recruiting prior to the SARS-CoV-2 pandemic. I'm going to skip ahead. I had included some of this stuff just in case people were interested in talking about any of the specifics, just because it's so topical. But I want to get to a couple of other topics before we end. So one is about reactogenicity of the vaccines. So a big issue with reactogenicity is you feel sick, you feel injection site pain, headache, fatigue, elevated temperature. All of this is your innate immune response, and potentially after the second dose, your adaptive immune response secreting cytokines that induce all of these feelings, because they're all associated with your immune system trying to mount a defense against a pathogen that's actually just not there. But it really reflects the strength of the adjuvant. And so this is this constant battle in vaccinology where if you put a stronger adjuvant in, you're going to get a much more robust immune response, but you're going to get all of these symptoms. If you put a very weak adjuvant in, or no adjuvant, as we do in the flu vaccine, you're going to get, people are not going to mind the vaccine as much, but you're also not going to get much of an immune response. And there's a lot of work to try to dial out reactogenicity while maintaining immunogenicity. But I'm somewhat of a skeptic that those two things can actually ever be separated from each other, because all of this is really downstream of your immune system doing what it does. It's the way that the immune system signals to itself that it needs to make an immune response. And I don't think we have the tools yet to divorce the innate immune response from the adaptive immune response to induce a strong protection. And then yesterday there were some questions about modeling how long protection lasts, either after infection or vaccination. And so these are some of the early data from the Moderna vaccine trials. And you see that there is this decline. You have this very early rise, and then this slow decline. But this this is to be expected somewhat with the biology of how the immune system works. You have this very early hyperproduction of antibodies, and then as the response converts into a memory immune response, you still maintain a baseline level of antibody production, but you're going to lose those antibodies from that early period of the response. These data, there's tons and tons of data like this that are actually measured in lots of people at very precise intervals. And I think it's you know, a field day for modelers, because we don't have this data routinely on any other vaccine that I'm aware of, and certainly not on the sheer numbers of individuals. The other issue that I wanted to get to that relates to thinking about how this is measured and the biology behind it is a recent report on the potential longevity of the immune response both to infection and to vaccination. Looking at the generation of long-lived memory plasma B cells. And so I just wanted to bring it back to the immunology, because it's important to think about, again, you can measure the immune response in many different forms. So these measures here are actually just measuring the soluble antibody in the blood and measuring that antibody over time. So that's something you can model, and it's something that you can determine the longevity of the immune response. You can also measure the cells that are making those antibodies. And so, as we've talked about, the antibodies get made from a naive B cell that gets activated by its antigen, that starts proliferating and then decides to start secreting antibody. But you also then have memory B cells that aren't secreting antibody, that are just going to activate more quickly if they see the antigen again and convert into cells that secrete antibody. And then you also have these long-lived cells that secrete antibody. So you have cells that are made very quickly that secrete antibody that are called short-lived plasma cells, and then these long-lived plasma cells. And the long-lived plasma cells have not been studied very much in many infections because they live in the bone marrow, and you have to get people to give you access to their bone marrow. But because it's SARS-CoV-2, people are starting to give people access to their bone marrow. And there was this paper just about a month ago that looked at antibody responses here. So this is measuring soluble antibody responses after infection, and then looked at the formation of long-lived plasma cells specific for SARS-CoV-2 in the bone marrow, and saw them produced at a very robust rate, comparable to what we see for influenza. And we know we generate long-lived memory to specific influenza strains. And I threw this in here because you're going to see this kind of data a lot. There's many applications of this technique, and this technique is called the ELISpot technique. And in this particular activation or implementation of it, what you have is you have a little membrane at the bottom of a well, a little piece of paper. You put your B cells from your bone marrow on that piece of paper, and you also have coated that piece of paper with SARS-CoV-2 antigen, in this case, spike. And then the B cells will start making antibodies, and they will bind to the spike in the well. And you add reagents then to read out the antibodies to spike in the well after you've washed the cells off. And those reagents turn color if they see spike antibodies bound to spike antigen in the bottom of the well. And then you get these spots, and each spot corresponds to one cell that was secreting antibodies, and you can count them up, and then you can quantify them and show that you're inducing this very nice, long-lived, robust SARS-CoV-2 response here. This is SARS-CoV-2 being compared to tetanus, which we all get primed and boosted with a lot. So look, these people are making many, many more SARS-CoV-2 antibodies. Oh, sorry, this is flu. This is SARS-CoV-2. We have tons and tons of flu. We have some tetanus, and then we have very good SARS-CoV-2 on the scale with tetanus. The RNA vaccines are also inducing robust T cell responses, which can also be measured by ELISpot, which is what was done here. And so you can see everybody's making both CD4 and CD8 T cell responses. Again, here they're stimulating, though, with large peptide pools. And we're probably massively underestimating the CD8 T cell response.